This summer, I had the honor of working in the gall lab. And before getting into the mechanisms of placental malaria, it's important to first understand the anatomy and the function of the placenta. So the placenta is the most crucial organ for the fetus during pregnancy, as it's responsible for passing nutrients, gas, and waste exchange between the mother and the fetus. The two main structures my research focused on were the placental villi. They're these finger-like projections originating from the fetal cryonic plate, and the intervillous space, which is the void between those placental villi containing maternal blood. Fetal Hofbauer cells, or HBCs, are fetal macrophages in the placental villi, and maternal intervillous monocytes, or MIMs, are maternal blood monocytes in the intervillous space. This here is just a picture of a cross-section showing the HBCs. Now, monocytes and macrophages play an important role in inflammatory responses. Oops, sorry. Oops, sorry, having technical difficulty. Are you guys able to hear me okay?
Yep, can hear you fine.
Okay. Sorry, my Siri started and I was unsure if it's cut off. Okay, so monocytes and macrophages play an important role in the inflammatory responses as they can polarize into either pro or anti-inflammatory activation states depending on environmental stimulus. Now, placental malaria is a parasitic infection transmitted through mosquito bite. When an infected mosquito bites a human, forms of the Plasmodium parasite travel through an individual's bloodstream to reach the liver and invade those red blood cells. Malaria is mainly seen in sub-Saharan African regions, and to understand the prevalence, statistics from the World Health Organization shows that 247 million cases were reported in 2021 and 619,000 resulted in death. Now, placental malaria is a complication of malaria pregnancies, and when infected red blood cells sequester in the intervillous space, those infected red blood cells can bind to the outer STB layer of the placental villi. It is diagnosed after birth by histological analysis of the placenta and is broadly divided into two categories of active and past PM. In active PM, we see presence of the parasitic infected red blood cells in the intervillous space, whereas in past PM, we cannot see those infected red blood cells, but only the hemozoam pigment, which is the waste product of the parasite. This first image here is of a normal placenta with a normal amount of red blood cells in the intervillous space. While over here is an image of an infected placenta, we can see the abundance and the intervillous space, as well as these infected red blood cells. PM can lead to poor adverse outcomes of fetal growth restriction, low birth weight, preterm birth, and miscarriage. The aim of my project was to identify the activation states of the MIMs and the HBCs to determine differences of active and past PM. We had 21 samples, 7 healthy controls, 7 active PM, and 7 past PM. We used an RNAscope, which is a commercial RNA in situ hybridization kit that enables the detection of mRNA molecules using gene-specific probes. They attach fluorescent dyes, and we can detect different markers using fluorescent microscopes. These were the 5 markers we used with their corresponding colors. And over here to the left is an example of the final image we got and what I scored. So we used a program called QPath that visualized each marker in separate channels. This white arrow here is showing the area that each channel is zoomed in on. And then from there, based on the location, whether in the placental villi or intervillous space, and the expression of the markers, we counted and categorized each macrophage. For our results, we counted monocytes and macrophages by their CD68 expression. For these 3 graphs, the cells in the placental villi represent the HBCs, while the cells in the intervillous space represent the MIMs. These charts are paired comparisons, so the connected dots are from the same sample. We found that in healthy controls and past PM, the density of HPCs was significantly higher compared to MIMs. So we see that over here in the healthy control as well, but this significance was not found in active PM. For these 2 figures here, they only represent the cells counted in the intervillous space, so just the MIMs. The CD163 is an anti-inflammatory macrophage marker. Therefore, the ones that are negative are considered as pro-inflammatory. So this left graph here. We found that active PM patients, these middle red dots for both graphs, had a higher density of both anti-inflammatory and pro-inflammatory MIMs. And finally, I would just like to thank everyone from the SSRP and the GAL Lab for everyone's support in this amazing experience. And thank you so much for listening.